Welcome to JMBE Live. This is an occasional webinar series featuring recently published curriculum articles, research articles, and tips and tools from the Journal of Microbiology and Biology Education. I'm Rachel Horak. I'm staff in the Education Department. It's my pleasure to welcome you today. Today we have a curriculum article, and I'm going to put the link to the paper, which is open access, into the chat shortly. This webinar is being recorded, and you'll be able to watch the recording and all recordings of other JMBE Lives on our YouTube playlist, and I'll put the link in the chat shortly. So with that, I'm turning it over to JMBE Editor-in-Chief, Stanley Maloy. Thank you for joining us today.
Welcome, everyone. It's really nice to have you here. I have to say, I really like this paper. It's nice for a couple of reasons. One is, I think, as in the title of their presentation, it reflects an old dogma, the central dogma of molecular biology, and how surprising it is that students are challenged with that dogma throughout their undergraduate education. And what they've done to try to figure out a new way to ingrain this concept in students' heads so they can really think about it in more meaningful ways. The authors that we're going to talk to today are Nancy Boury and Greg Phillips. This is also a story that I really like because Nancy is an education researcher, does some nice basic research too, but a bona fide education researcher. And Greg is really a basic researcher in microbiology. And so this came about from them talking to each other and figuring out ways that they can address this very important problem that students need to understand. So it's going to be a fun tour that we go on today, and I'll turn it over to you, Nancy and Greg.
Hello, all. I'm Nancy Boury, and this paper is the product of quite a few hallway chats between Greg Phillips and myself. And if you want to take over for a second there, Greg.
Yeah, I guess we should say the hallway chats are not occurring as frequently now because I moved to the University of Georgia, so not in the same hallway anymore.
But yes, sadly.
Thank you. Several years at Iowa State University, and Nancy and I were actually on different floors, but I would come by to use the copy machine or something, and we'd strike up a conversation. And we won't say how long ago those conversations started, but they did result in us actually trying some ideas. Nancy had some nice ideas on assessment, and we added to an experimental program that we had going, both in my own research lab and kind of brought that into the teaching lab. So that's a little bit of the background, the genesis of our project here.
Yep. And coincidentally, we came up with the title of Teaching an Old Dogma, New Tricks independently, but it was the same. So I thought that was hilarious. So
yeah, like an independent mutations occurring in two different cultures.
Something like that.
Yeah.
So kind of moving on to what we were doing and why. The first thing I want to kind of point out is the illustrations on this paper were done as a independent study project for our biological and pre-medical illustration program. I often have students that sadly they will come to my class, they'll see me drawing on the chalkboard or whiteboard, and they will come in after class and say, do you need help with your drawings? And that's actually how I met Sparrow Hartman, and they were very happy to help me out with these and wanted a project for their independent study. So that's how that happened. But so the central dogma of molecular biology, from DNA to RNA to protein, and then DNA replication and reverse transcription, all of that really is foundational to molecular biology. But well, students can often at their senior level can, recite DNA, RNA, protein. When you ask them things like, what does a stop codon stop? As part of some other research I was doing, I actually asked that question, and 50% of over 200 students in their sophomore year or better, 50% said transcription and 50% said translation. That was the kind of the breakdown, which, it is a common problem at the fine grained details. So that was part of the reason we wanted to do this.
So Nancy, don't you think too that the simplicity of that central dogma figure is a little bit confusing, because when you'd ask students to draw this out, RNA, DNA, protein, they can almost always do it.
Mm-hmm.
They get the arrows right and such, but the details of what's included in those arrows is really what we thought was lacking. And then when you go from that RNA to protein, the translation, right? That's complicated machinery, cellular machinery, a lot of moving parts there, and I think that's where we really saw a breakdown in comprehension.
Agreed. And just distinguishing all of the pieces, and the idea that not everything that's transcribed is translated. So one of the things I kind of wanted to do as an interactive was to ask the group, what are some of the things that your students struggle with the most? And being as we're post-COVID here, we've kind of done a lot of open access things where you do a waterfall response, where you type in what you think your students are struggling with the most. You type your response in, but don't hit enter. And then, I'm going to wait basically a minute and see, have this all hit at once as a waterfall. So if you could type it in the chat, and we'll wait a minute and have everybody go at once.
Yeah.
Okay, I'm seeing terminology, DNA replication, translation, details, promoters versus ORI versus start codon.
Mm-hmm.
So what I'm seeing from a thematic, from the responses, the themes I'm seeing really go down to what the details are. So as Greg was saying, they can recite DNA, RNA, protein, but do they understand what that really means? And there are a lot of moving parts going on in that system, and being able to understand how those pieces interrelate with one another is important to molecular biology.
Yeah, so I can fill in a little history here, a little background, and likely many of you are familiar with GFP. You've used plasmids that express fluorescent proteins in transformation experiments. But for several years now, it's been known that there are proteins that are similar to structure, and we'll show you that in just a second here, to GFP, but rather than emitting fluorescent light, they're actually absorbing light, and then your eye can pick up the wavelengths that is not absorbed. And these are found very abundantly in marine invertebrates, so corals and sea anemones, and just the abundance of marine life that gives off these colors. And more researchers have been looking into that by actually identifying and cloning the genes that express these so-called chromogenic proteins. We call it CP for short, and I'll continue that designation. And so unfortunately, now that we're in a area, I guess fortunately or unfortunately, an area of synthetic biology, is that we did not have to go to any exotic locations, isolate coral, and extract the DNA and clone the genes. Rather, we rely on other people who have done that. And there's an example of a paper here. There are many other papers. Many people have done this. And so we were able just to find that DNA sequence that had been published, and then order the sequence, type in the ACTG combination, order that up from IDT. And a couple of weeks later, we would get a little tube, and we would take that DNA and clone it and express it. And just shown here on the slide, this striking resemblance between the CP that's nice and purple, and this is in a species of Acropora. But then when we take that same gene, that same CP gene, and express in E. coli, essentially those E. coli turn purple as well. And so we've been using this for several years to dissect some of the other steps of the central dogma. So, for example, we can express the CP gene, therefore revealing this phenotype under different types of promoters. And I'll show you an example of that coming up. We also did mutagenesis. And so that had been a part of our class effort for several semesters before Nancy and I started with this. So this slide just shows a little bit more detail of how we can use this relatively simple gene. It's a very small gene that the protein is actually less than an average size E. coli protein. A lot of people have worked on it, like I said. So on the left there is just the structure. And if you've seen those structural figures of GFP, essentially this looks just like GFP. It's a barrel structure, and then the action, so to speak, is in the center of that barrel. And that, by that I mean the color is emanated from certain amino acid combination in the center, just like GFP fluoresces. This one is absorbing light. So a lot more understanding reveal that amino acid 62 is very important for producing the color of this CP. And so we and others have decided, well, can we alter that amino acid and produce other colors? And in general, you can, and there's different examples there. So the normal amino acid at position 62 is a glutamine, abbreviated by the N. And if you change that to just using DNA technology, just doing site-directed mutagenesis, other ways of doing that, to a leucine or a methionine or glycine, you actually get very different colors generated. So the little plate there is just a mixed culture where we have done a randomized mutagenesis of the DNA encoding the amino acid at 62. And when you plate those out, you get quite a spectrum of color there. Not every amino acid that you replace in 62 is actually functional. And that could be interesting for some other structure function analysis as well. So we were doing this for a while, changing the color, i.e. the phenotype of our E. coli, just by a simple mutagenesis of this one position in the CP gene. So I think in the next slide, is where we started after the conversations that we had with Nancy, that started thinking about, can we use this for a specific teaching tool?
Right. But before you do the teaching tool, you need to know about what your learning objectives will be. You're getting so in good backwards design format, we're looking at, we've got these tools. What do we want to do to teach with this really cool system that Greg had? How can we design an exercise that's going to dig deeper into some of these details? So by changing the DNA and getting a different phenotype, that really talks about mutagenesis and the role of DNA sequence into the message and connecting genotype to phenotype. But then the next thing was, can we change something else and still get the change in phenotype? So our student learning objectives for this project really were for the students to be able to identify the features and the structural components, all of those complicated moving parts we talked about earlier, and differentiate what's going on in translation from what's going on in replication, like the difference between a genome change and the change in the DNA versus a change that we've had with translation. So we're not changing the CP gene, we're changing something else. And then so differentiating between transcription and replication, and then looking at suppression of a mutation, because we started out with this project, the CP gene that goes into these new bacteria that Greg will talk about here in a second. The CP gene started out as with a nonsense mutation at that important 62 amino acid position. And then, we want students to be able to critically evaluate their results and then explain their results. So not just get the data, but be able to explain it in terms of the molecular mechanisms behind it. So how are we getting a changed phenotype without actually changing the plasmid that's generating this phenotype, like the mutated CP gene? Okay, and we align these learning objectives with what ASM, the fundamental statements with ASM's curriculum guidelines, and GSA, or the Genetic Society of America's educational framework, because there was a fair amount of overlap in that both of them are based on vision and change. And so this was the slide that I think you were going for, Greg.
Yeah, so we had successfully, we believe successfully incorporated transcription into the CP model system, because again, we could turn it on and off by different promoters. And we had done replication. These are plasmid-based systems, and we could do mutagenesis, showing how DNA, once it's replicated, then segregates out new genotypes reflected by new phenotypes. But that last link of the central dogma, the translation, how could we effectively show that using these dramatic phenotypes of the CP gene? And that's, we were talking offline before we started. And I think Stanley and I both just have always had an interest in this idea of genetic suppression. It's just an amazing system, and the individuals that figured this out early on in bacterial genetics, just incredible insight. And we were thinking, how can you use that? And Stanley probably also, we're both really entrenched in the lac operon system, right? And how suppression was, the discovery of it and characterization of it was in part linked to lac operon. So we kind of think of this as an updated version of the lac operon for teaching students. And so the basic idea of genetic suppression, right, as Nancy said, is the mutations, the alterations in function, not associated with the CP gene, but rather components of the translational machinery, specifically tRNAs, those linking molecules that link up the information in the messenger RNA with the eventual protein product. So many years ago, it was discovered that you can get mutations in these tRNA genes, right? They're encoded by the DNA. They're not translated. They're encoded by the genomic DNA, and they're prone to mutagenesis as well. And occasionally, mutations can occur within a gene that encodes a tRNA such that the specificity of that tRNA is changed, and that mutation is right in that region that encodes the anticodon loop. So right away, you're having to kind of, to understand this, the student has to delve into the translational process and go through these figures or look at videos and how the machinery all connects together. So we reason that if we could just introduce then the CP gene that had been essentially disabled by putting in a nonsense mutation, a specific nonsense mutation, TAG in the genome, or in the DNA, and UAG in the actual codon. So that's a specific kind of nonsense mutation called an amber mutation. I always wondered what that meant and did a little digging in history. And apparently these are sort of play on words in German. They're supposed to be kind of jokes, but they're not very funny. So we just call them ambers, right? And so there's a couple others, but the amber works really well for this suppressor experiment. So we just took our same platform plasmid, it's CP, it's actually expressed by control of rhamnose. So you actually add the sugar to the culture media, and that's what turns on the gene. But the gene doesn't work because we've introduced this amber stop codon in the same position of amino acid 62. So within the experiment is really very simple. It's to take this plasmid and transform it into multiple E. coli strains, each of which has a different mutation in a different tRNA gene, meaning they're different suppressor strains. And all that is summarized here. A lot of information in this slide here. But basically, we selected 5 E. coli strains, and 4 of them are suppressors. They have these altered tRNA genes, and then the 5th one there is this wild type. So it's a normal E. coli. It doesn't recognize an amber mutation as being any different. It treats it as a stop codon. Well, we were fortunate because strain number 1 there actually is a suppressor, and the tRNA that's actually altered is for glutamine, which, as I just said, was the wild type amino acid at position 62. So indeed, if you restore glutamine to position 62, both in the little colony picture there, and we'll see some more examples later, but in broth culture, the purple CP phenotype is restored, right? You're just replacing the normal amino acid there, but you're doing it again through the translational process, not by changing the CP gene sequence. Then we had a couple surprises that whenever you would insert a serine at that position, strain 2, the color would change to a different color, kind of a salmon phenotype. Strain 4 inserts a leucine, and we've changed the color yet again to a magenta phenotype. And interestingly, and students have to explain this a little bit, strain 3 inserts a tyrosine, but there's no color. So we did a little bit fewer and more experiments to show, yes, indeed, that the tyrosine is actually suppressing, but that amino acid just won't work at position 62. So there's a future there if you want to look at structure function, how certain amino acids will work to generate a chromogenic protein and others will not. So we're a little bit fortunate here that we were able to have everything from wild type to non-functional just with transforming these 4 different strains, and then the 5th strain just serves as our control. And we find that that's helpful too, to get students to think about controls, about how you can do a comparison with your results, and that can give some meaningful results.
And this is this was the diagram that the students were given, just to show this. They did the same plasmid into all of these different strains, and they didn't get the expected results piece to it. We just put this in here as this is what it looks in live. For live. But we did, from an educational research perspective, or to publish this, we wanted to see, does it really work? So we built a pre-lab quiz. I talked to Dina Newman and said, hey, it seems you did a concept inventory for the Central Dogma. Do you have those questions? And she gave me the concept inventory. I pulled out the questions that were specific to prokaryotes, because the Central Dogma concept inventory is fairly eukaryote forward. So some of the things that would not be appropriate, like mRNA splicing and other things like that. I pulled those questions out, and then we also asked several questions about suppression and several questions about how phenotype or genotype and phenotype are related, as well as just the Central Dogma question. So they got a pre-lab and then they got a post-lab, and we did this for several semesters because this is a lab course. And Greg, what was your average number of students in that lab?
We ran typically 16 to 18 students per semester.
Yeah, so it took a couple of semesters to get enough students to really get the data we needed to analyze. So just the whole point of this is that the students were transforming the plasmid with this CP gene with the nonsense mutation at that important amino acid position of 62, and then they put it just into different strains. So the same plasmid into different test tubes. And I think having them do that hands-on, it's the same plasmid, really did help.
And we had done some previous work with transformation, so they kind of had that technique down to ensure that it worked with every strain.
Yep. And this is the Gene Eat Wow slide.
So this is just a summary of maybe some offshoot exercises that we did. So on the left there is just that control, that regulation. We can turn on and off the color phenotypes simply by adding a rhamnose to the cultures. In this example, of course, these are on agar plates. The students just spread the rhamnose inducer onto the plates, and then they can observe not only the phenotypes in the suppressor strains, but showing you have to turn the gene on for this whole phenotype to be realized, and you turn that on by rhamnose. Since it's a microbiology lab that we were teaching, we had a couple other ways in which the students could culture the bacteria. So shown there are the broth cultures, the 4 strains without the control. So the color does develop in broth culture as well. And then kind of for their notebooks and for future reference, we would have them remove some cells, simply pellet them in microcentrifuge tubes. And that really seemed to concentrate the cells, of course, and concentrate that color. So we use a digital lab notebook, so they could take pictures of their cultures, their plates, and then upload those to their notebook. And then as they did their lab reports, they could take those figures and add figure legends and do a more formal report.
And so the other part for this was, they had the lab report, which was mostly the discussion that you would write as if this was a paper to be published in an ASM journal, because we want to keep them in a consistent journal format. But these were the questions as they were finishing up their lab, what that the students were really asking them. Well, we were asking them and they were answering, but as a large group discussion at the end of the lab, and then they took that data back to their dorm rooms or wherever they were writing their lab reports. But the pieces that we wanted to really look at were, after transforming these, we saw different phenotypes. What's going on here? So are the DNA sequences in the plasmids randomly changing? And the answer to that is no, but we wanted them to be able to not just know, but what's going on here. And then the other thing is, suppression is a mutation, but it's a mutation in tRNAs. So we want them to recognize that the tRNAs are transcribed but not translated. And then the other piece to that is, as they're transcribed, there's another process that has to go on, that amino acids have to be added, so that charging of the tRNAs has to happen. And so we wanted to those fine details as well. And then the last thing is the difference between reversion and suppression, because again, we want them to look at the central dogma at several different levels, but also reflect on and state and explain to us that they understand that a lot of things are going on here, and you can change a phenotype at multiple different levels and the differences between them. So most of this is, again, the molecular mechanisms behind this. But this was something that Greg was leaning much more than I was. But looking at some of the lab reports from the data analysis perspective, I think a lot of times they really did come through like with these molecular mechanisms in mind. And then, we said they did a pre-test and a post-test, which they did. And they, these are juniors and seniors. And so their average score that, it was the concept inventory total score was 67. It wasn't 67 credits or questions, but that was the total score. And the pre and post, we did get modest gains, but it went from like a 75% to a 79 to 80%, if you round up, because I think it was 79%. We did have quite a bit of variability in the data, but is this statistically significantly different? Yes, the p-value is quite low. Normalized learning gains were about 14%, which is, again, moderate gains in their learning. And this was across several different semesters, including the spring 20 semester, which it had its own wonderful data set there. And if you're wondering, normalized learning gains measure out of how much the students could have learned, they did learn. And the way you calculate that is the simple post minus pre, but then you take whatever the 100% score was, in this case it's 67, minus whatever each student's pre-score was if you do it by student, or the average pre-score if you're doing it by averages. But this gives you the ability to compare scores that students that were low scoring, how much did they gain, versus you get a bit of diminishing returns if a student starts out in like the 90% range. This kind of bring some of that crazy wild data into control. It controls for that a little bit, is what normalized learning gains do. So, this one is kind of an open. I'm not going to do the waterfall again, but since you have the chat, how would you use these tools? And these are all available through AdGene, thanks to Greg, and Greg's graduate students posted them right as we were submitting this paper.
And I'll add too that this is certainly not a unique tool to our interest. I mean, our classes, but so there are other chromogenic proteins. There's a couple of kits that other people have deposited, not suppressors, but kits that express different variants of the CP gene. They're available on AdGene as well. So I can show you a little bit when we're done here, Nancy. I can show you a little bit about what else we are doing with this and what we're hoping to do at the University of Georgia here.
Amazing. Not seeing a lot of, I gotta look at the chat. Oh, the amber mutation. I'm gonna have to look that up when I'm not doing this. So some of the ideas were, the idea of plus and minus rhamnos. So you're looking at transcriptional control, and the fact that you can do kits that have students do the beta barrel, like cut out and fold the beta barrel. And that is something that Greg's students were doing earlier. I can't tell you the number of students I've seen in the break room folding and taping their beta barrels for CP genes.
That started in the COVID year, and we would actually actually it was a high school group, and we sent out these pretty good cardstock models, and they would cut them out, and they're really well designed, and you could fold up your own little beta barrel, and then when you were done, I think they would be an appropriate ornament for a Christmas tree as well.
Well, they were pretty cute. And then the idea of mutagenesis. I really love your multi-plate that you made, where you're like, yeah, we're going to change that amino acid.
We were starting to add sequencing now, so you could have a mutation. You could isolate it based on the color, and now with sequencing costs what they are, you could actually just sequence the whole plasmid. So students could learn about how the different elements of the plasmid work, and then identify the basically single base pair change that occurs in many of these sequence variants. We talked about protein structure function. And then with a little bit more advanced course, right, these CP variants could be incorporated to generate biosensors, for example. So you could hook the CP gene up to a different promoter, one for let's say that's turned on in the presence of an aromatic compound, and then in the presence of that compound in the soil or whatever, you would get a color change as kind of a harbinger that this compound was in the environment. And I'm sure you all can come up with many more ideas of how these resources could be used, but these are the ones that we are pursuing now.
I was actually thinking about these and your multicolored plate from the recent microedu conversation about how to get students to know that they've isolated, like to streak for isolation. If you gave them a mixed E. coli that like all the different colonies would be very different colors or lots of different colors, you could tell if they were isolated.
Right. Just have to add rhamnos.
Right. So, so the other thing, this is supposed to be a conversation, so questions. And as we're asking, pulling out questions, I'm going to stop sharing. But the one thing I did want to say is, the students in our Micro 440 class really did a lot of work and really showed us how their understanding works and what we can do to help them. So, shout out to the students. Also, undergraduate student workers, mostly in Greg's lab, a couple in my, and then we had a graduate student and that was also I'm listing the people that weren't on the paper, although Rachel was on the paper. And then March is a postdoc that was in my lab, and Sparrow is a BPMI student. And it really did take a whole bunch of people to put this together because just like translation, there were a lot of moving parts.
Thanks very much. Thanks to both of you. That was really great. And in fact, I think that one of the things you started with, Nancy, is mind-blowing to me, that 50% of seniors could not tell you what a stop codon stops. That says a lot about the need for us to think hard about doing this in a better way. You made two other points in the paper that I think are really valuable, and I think it ties to why that's 50%, right? So one thing you said was giving students correct factual information on biological processes is not effective at addressing misconceptions. You cited a very interesting paper there, but I think that's something the audience might really be interested in, because just doing it in lecture doesn't matter. And this is where I think active learning in the labs, the creative labs that you guys built, are really valuable. But it also emphasizes a point you made in your paper, is that cookbook labs don't do it. It's easy to get in on a cookbook lab and do what you got to do and get out with limited new understanding of complexity, right? So I really like the fact that this lab, it isn't just a lab, it's an active learning exercise, right? So those are things that are really cool. In your paper, for those in the audience who have a picture of this, in your paper, you show a variety of changes at that amber mutation, and one of them inserts a tyrosine. And that insertion of a tyrosine looks exactly like a stop codon. It results in a non-functional protein. So how do the students deal with that kind of bit of confusion?
Not well sometimes. They all expect, I think it goes back to your point, Stanley, of expecting a cookbook lab where, of course, you're going to get something happen. And then when nothing happens, there's a little bit of confusion there. It seems like everything should give me a different color, right? Because we planned it that way. So in a more of an advanced lab, we only did this once, but we actually then ran a SDS page gel and show that, yes, the protein is there. You can see the band, on the gel if you do it carefully enough. So suppression is working, right? The amino acid is being inserted. There is a polypeptide made. And then that, I think, helped a little bit to get to the point of, oh, it's a non-functional protein rather than a truncated protein.
I think that's really a good point, because I can as a teacher, I can see the confusion that can rise very readily, right? And make people go.
Or why would we give them a strain where there was no cool new color?
Right.
And then that brings in also the structure, is showing, well, not every amino acid works in every position.
Right. I mean, I think that actually one of the papers that you cite, I believe, in your references, is about the value of experiments that don't work the way you expect it. And I think that this is really a great example of catalyzing that thinking. One of the other things that I thought was really impressive about this paper was, Nancy, you did a great job of taking us through that table 2, where you talked about the increase in understanding tied to this data. And if you look at the data there, if you don't apply that kind of analysis that you do, you wouldn't have realized about the amount of change that this really invokes. So the mean numbers go from 50 to 52.2, but the fact that the learning goes up by 14% is really valuable, right? I mean,
Yeah, we're leaning into the physics education folks. Dr. Hake, about maybe 20 years ago, really was the champion for normalized learning gains. And I went to a webinar that a bunch of physics folks were putting on years ago, and I became a true believer because, just as you're saying, you can really mask changes from students because the difference between a student getting 95% to like 97%, that's very, very small in your data set. But if you think about that 95% being they could only gain 5% more, and they gained half of what they possibly mathematically could, that's a big that's a big change. And so you need to really think about not just what the raw numbers are, but what they could have learned and what they did. So
the other thing that you mentioned in your talk, although it's not in this paper, but that you can run the normalized learning gains as individuals as well. So presumably if you'd done something like this early in the semester and you looked at the individuals, you might be able to target some people that needed additional interventions to get to the desired level of achievement, right?
Correct. So I mean,
yeah. I think that can be a valuable point of, where both the averaging of all the data across the students is really valuable, but I think you may be able to learn some real important things by looking at particular individuals, and potentially you could even go back and say, oh, what courses does this relate to courses that they had previously, or and maybe help you change the education system more broadly, right?
Well, I teach a lot of the general courses, and I always give the normalized learning gains for the class, like the entire class, for general bio and general genetics and general micro that I teach. I give those numbers to the chair and whoever is asking for it, because I want them to be able to see, and I want to be able to see what's going on from what the students could have learned that they did. I mean, you could do just the raw data because from a general class, you usually get much bigger. The gaps are a lot bigger, but for the advanced classes, you're usually going to see a smaller, you have smaller space to gain because they're already starting so high.
So along those lines, one of the nice things you did in your appendices, you provide all the details. So if anyone in the audience wants to reproduce this, they have everything that they need to do it. Including where to get strains and so on. But one of the things that I think is something worth discussing is there are always some challenges with teaching an advanced class like this, right? And I wonder if you could comment on, like, Greg, what do you think the challenges are for implementing a class at this level for seniors?
Yeah, so I guess a little bit of my surprise. So this is actually just a part of a larger advanced microbiology class, and our module, our sections, 5 weeks intensive on bacterial genetics. But I guess to my surprise, it shouldn't be, is just the diversity of backgrounds, even within a microbiology major. So at a large state public institution like Iowa State, you had everyone who was interested in important aspects of microbiology, from food safety to environmental to those looking at professional, pre-med, pre-vet. So even within a pretty specific major, my challenge has always been to get everyone on the same page, have the same learning outcome and learning interest. And I realize this may be hard for you to understand too as well, Stanley, but not everyone was really into bacterial genetics. I don't understand that myself, but it was true. And so how do you just tell people, get them excited about the potential of this type of technology when they're going to go to vet school or whatever? And so we try to tie that in with biotechnology, with synthetic biology, of development of new drugs, and maybe that helped. We have no statistics on that, but more of a challenge, I guess, in diversity than I originally thought.
One of the, as an aside, and we'll come back to this, but what got me into being interested in bacterial genetics was actually taking a class my senior year that was really focused on thinking, not memorizing, but really thinking about things in the way that you in this lab have made people think about things. And I think once you start to do that, thinking can be really addictive, right? So
let me think about that.
But what I was wondering if you might say something about is, for many people at different institutions, having a class of 16 or a lab with 16 or 18 students might be cost prohibitive, right? Especially if you invoke the amount of additional TA work that might be required and so on. And so I wondered if you might say something about scale up. Do you think this is scalable?
Yeah, so specifically for the suppressor experiment, it's plates and toothpicks. Again, going back to your comment about thinking, a lot of the value of it, I think, is in the mental processing once you see the phenotypes and making sense of it. So we're not doing any, we can add the sequencing, you can add the site-directed mutagenesis and all that, but we think we're getting a lot of mileage, so to speak, out of the phenotypes, just looking at the plates, the agar plates, and restreaking with toothpicks. As our former colleague has said, toothpicks and logic is a big part of bacterial genetics.
Yeah. But by the way, Greg is referring, if any of you know, to Tom Sahabi, who influenced both of us in our careers. If anyone in the audience has any questions, please post them in questions and answers, and we'll get to your questions. I'm happy to chat with Nancy and Greg for a long time, but we'd love to get your questions as well. So please post the questions. So one of the other things that I think is really nice about this study is that it really brought in your expertise, Nancy, right? So fundamentally, if Greg had done this on his own, he probably would not have had the ability to parse that data in a way that would lead to a publishable paper. People would say, right. Like there's any difference, right? And so, I think that type of collaboration can be so valuable. And I wonder if you could just say a little more about it, Nancy.
We were only half joking when we said this project came out of discussions, hallway discussions. And I think if I remember correctly, and correct me if I'm wrong, Greg, this might have been one of those, you had really spiffy looking plates that you were transporting like through the hallways. And I'm like, those are really spiffy looking plates. What are you doing? And he explained it. And I was like, that sounds amazing. Do you want to think about like, what are your learning objectives? And I got a learning objectives. What? And I'm like, we'll work on that. And so we ended up talking about this for several semesters. And, like the first year we were, I kind of interviewed Greg as to, what is it you're doing? What thought processes do you want the students to have? And we really worked on refining pieces of the exercise, and that started with the learning objectives, but it then talked about what the discussion questions would be. And then after, trialing it once or twice, then we changed the learning objectives a little because we realized, so there was a bit of a give and take there. And then finally, I think we got it to a point where we're like, okay, that's great. Now we need better pictures. And that was about the same time that I embarrassed myself with my drawing in class, and Sparrow jumped up and they were amazing and said that they needed as part of their bachelor's degree in BPMI, they needed a portfolio that included working with clients. So I became this BPMI student's client, And then we got pretty pictures. And we had Marcia, who was a postdoc just for a year for me. She's now at UTEP, and she has expertise in some of the data handling. And so that was, gathering enough data. So it was a very lots of people that contributed to this. But I agree with you, Stanley, that it's amazing to be able to work with faculty that are doing really, really innovative things, and then just tightening up some learning objectives and just doing some things that put it into the format that it is publishable. Because it was a great idea to start with. All I did was organize it a little better. So.
Yeah, and I'll just add, I wasn't making light of the importance of learning objectives. Clearly, I knew about them and included them, but working with Nancy really reinforced not only the importance of them, but then how they're formulated, so that they're measurable, and then students can look at them and say, okay, I see what I'm trying to accomplish. So I think that partnership, definitely helped me in teaching experiences beyond just this lab exercise.
Yeah, I totally get it, and I understand where you're coming from. I think that is a common refrain from people who haven't really looked at the real benefits of learning.
Nancy taught me how to write good ones, not just we're going to learn something new,
yes,
rather than having them be specific and measurable.
Right. We'll go to questions, but let me just say about learning objectives. The other thing that you brought up, Nancy, was reverse design and how valuable learning objectives are for that. And so I think that's another key thing here. So one of the questions that was posted in the chat is, do you think that sophomores could successfully do this in a microbiology course if the only course that had with Central Dogma before was an introductory biology course?
So I think that's a great question. And I think the answer is yes. And I could actually think of advantages of doing it earlier. So we're doing it kind of late in the process here. Think how much more we could build from the understanding the basics. If they could learn as a sophomore, we could do much more in our labs by the time they are a senior. We could be doing more of the synthetic biology and bringing in some more sophisticated elements. So yeah, given the relative lack of technical sophistication needed to do this experiment, I think that's a great idea.
I'll just add for the person who asked the question, if you do it, be sure to get data on it, because you're looking at a different group of students. So you might see an even greater change in the relative learning. So
that's a good point.
So another question is, have you thought about a different type of suppressor experiment that would work? So for example, with interaction suppressors or missin suppressor, other types of suppressors maybe.
Yeah, so we've not done this for a lab class, but you could split these barrel structures. You can split GFP and then actually bring it back together within the cell by having interactive components. And we've not tried that for CP. I suspect it would work. It would diminish the output. Fluorescence is always going to be more sensitive. But so, yeah, I definitely think there's more that we can do. And we didn't explain all that we've done with the mutagenesis. So you can do random mutagenesis at that 62 position. You can do targeted mutagenesis, and then you can expand out because there are other sites that form that, other amino acids that form that chromophore. So yeah, I think the potential is not fully met in our simple experiments here.
That's the idea of a paper, to catalyze more experiments.
Yeah.
So then another question is, do you think that this could be practical in clinical microbiology situations? For example, demonstrating aspects of antibiotic resistance or inducible resistance.
So speaking of, yes, absolutely. Again, going back to this diversity of students that we typically have in this microbiology class. So some of those students would be interested in either professional, pre-med, pre-vet, or clinical type position. So we would do antibiotic disc testing and use the bacteria that were expressing these CP phenotypes, not just the suppressors, but really vibrant colored ones. And so you get this really distinct zone of inhibition between where you have killing, and then here's this purple lawn of bacteria that starts to come up. Another way that we've done this a little bit is actually in mixed cultures, right? So you could mix a sensitive with a resistant microorganism, mix them in culture, add an antibiotic, apply selection, and then after a period of growth, plate them out and visually see the purple one versus the magenta one or whatever. And then the magenta one goes away because of the presence of the antibiotic.
We have a few more questions, but in fact, we're coming up on the hour, and we have a few final comments that need to be made before we do it. But I'd like to say thank you to both of you. I think that clearly you've catalyzed a lot of people to think about how they could use this in their own lab. And I hope that you've also helped people think about ways that they can really use education research to study these processes they do, so that there will be evidence that they're valuable and other people will incorporate them into their own teaching. So
yes, and please reach out if there are other questions that we didn't get to. I'd be glad to continue the conversation.
Yep, ditto.
Okay, so since we do have a time frame that we have to finish this, I'd suggest if you have some other questions, or if you would like to incorporate this into your teaching, and you should contact Nancy and Greg and talk to them about this. And they're both very nice people. I'm sure they'll be happy to give you advice. So thank you very much. And I'm going to turn it over to you, Rachel.
Thank you, Stanley. That was great. It's really exciting to see these learning gains for such an important topic, such as Central Dogma, that all of our microbiology students need to know. So into the chat, I just posted some links. One is that you should go register yourself for 3 more JMBE Lives. We have one Tuesday, March 19th, and this is a very special episode. It'll be at 9 p.m. Eastern time. This will be the first time that we've hosted Asia-based presenters for JMBE Live, and that's why it's at 9 p.m. Eastern. The speakers are from Singapore. And then there will be another one May 3rd, and then another one May 17th. And do check the times when you register, because they're all a little bit different. And they're all free, and they're all open access articles. So go register yourself and continue learning with JMBE for the rest of the spring term. The next is a link to catch up on past JMBE Live episodes. We've made them all freely available worldwide on our JMBE YouTube playlist. So go check that out. I think we have about 30 videos right now. Some of them were filmed at ASM Micro meeting, and some of them have been filmed on this Zoom format throughout the last 4 years. So take a look, and we hope to see you at a future ASM event. Thanks for joining us today. And the contact information for our speakers can be found in their journal article. Take care, everybody. See you at the next JMBE Live.